Good morning. I'm Carl Alston from the Leukemia Research Foundation, and welcome to our Blood Cancer Awareness Month live stream conversation with Dr. George Souroullas, Ph.D., who is an assistant professor in the Department of Medicine, Oncology Division, Molecular Oncology, at the Washington University School of Medicine in St. Louis. Good morning to you, Doctor.
Good morning.
How are you today?
Very good, thank you.
Well, you are the recipient of a $100,000 grant, research grant from the Leukemia Research Foundation. So congratulations on that too. That's a big achievement. That'll move things along a little bit for you, I would think.
Yes, thank you, and that will definitely help us.
Cool. So Dr. Souroullas, the project that we are funding of yours right now is titled Understanding the Oncogenic Mechanisms and Chromatin Interactions of EZH2. So can you tell us, what does that mean? And tell us a little bit about your project and what you hope to learn from it.
Absolutely. Well, first of all, Carl, thanks for setting this up. I really appreciate it. I think we do need a channel to explain what we do to everybody else, and especially to the foundations that give us the funding. So what does that title mean? People probably have heard of the DNA. So the DNA is what codes everything that the cell makes. And many times in diseases, and especially cancer, the DNA code doesn't do quite the right job. So there's these things called mutations. So there's like errors in the DNA code that will code for, let's say, a bad protein, or a protein is going to do something that's not supposed to be doing. A few years ago, there was this huge project that was funded by the NIH and other sources called the Cancer Genome Atlas, that identified a lot of these errors in the DNA code in a lot of different kinds of cancers. When I got into this field, I spent a lot of time looking at that data, and another group had identified specific mutations in this gene. The gene is called EZH2. And these mutations were very frequent in a subtype of B-cell lymphoma and also some kinds of leukemias. So we wanted to understand what those genetic mutations did. Now, what is this gene? What is EZH2? So EZH2 is a gene that modifies what's called histone proteins. So what are histone proteins? So I mentioned DNA. So the DNA it's a very, very long molecule, and it's very highly compacted within the cell. Now, to achieve that compaction, the DNA is wrapped around these histone proteins. So it's like beads on a string. And these proteins that the DNA is wrapped around on, they can be chemically modified. And these chemical modifications on the histones regulate which genes can be turned on and which genes can be turned off. So mutations in genes that regulate this process are really important, because they regulate the entire genome, your entire genetic code. And this is what we refer to as epigenetics, or in this case, very specific chromatin modifications. So what this project is aiming to do is to understand how mutations in this gene, EZH2, result in the development of cancer. So the fact that these chemical modifications can happen across the whole genome makes it very hard to pinpoint exactly where the problem is, because they can regulate the expression of hundreds or even thousands of genes. So what we really want to do is understand what those mechanisms are, so when we are trying to design therapeutic strategies, we can be more specific and more effective. I hope that makes sense.
Oh, sure. That makes a little bit of sense. So what you're trying to do is sort of understand how EZH2 works and to what degree it works, and then try to, in the long run, try to find some treatments that impact specific, molecules or whatever to, impact them and stop tumor progression or stop tumors from being able to grow, basically.
Yeah,
right?
and all of these genes and proteins in the cell, they work in very fine-tuned networks, and trying to figure out where the problem in the network is, it's not easy, but it's important if you want to intervene in a way that's not too toxic and it's most effective.
Right. Okay. Well, that's understandable. You want to go right to the source, and that's also part of what makes treatment of blood cancers so personal, or what could be so personal. So what, if you have two people who have the same type of leukemia, for example, and they have the same, oncogenic problem, the treatment for that person, for one person, might be completely different from the other person when they have, like, on paper, the same thing. So
correct. So they might have the same type of leukemia, the same type of cancer, but if the leukemia is caused through a different mechanism, then they will not necessarily respond the same way to the same drug. So you try to tailor the treatment to their cancer's genetic profile. Right. So a person with an EZH2 mutation, for example, could be more a treatment, a small molecule that would target EZH2 will be more effective for that person, in theory, at least.
Yeah. So, okay, if you achieve what you hope to achieve with this test, or with this project, how will that change everything in the blood cancer world? Or what part of A will become part of B, I guess?
Well, these mutations are pretty high frequency. Like the 25% of germinal center diffuse large B-cell lymphoma patients have these mutations. So if we understand the mechanisms of EZH2, that would be very impactful, especially for the patients that have these mutations. And we've kind of touched on personalized medicine, right? Giving the patient a drug that's better suited for their genetic profile. But even within those EZH2 mutated patients, they're not going to respond the same way because of the complexity of the epigenome and the chromatin regulation. So in fact, there is one FDA approved EZH2 inhibitor that has been tested in clinical trials, and it has been effective. But a large number of patients that don't respond to that drug, even though it's supposed to be designed specifically for those mutations and those in those patients. So that means that we don't really have a complete understanding of what those mutations do, or what's downstream. What are the downstream pathways that mediate the development and maintenance of this cancer growth? So hopefully through these projects, we can understand these mechanisms and help us find more specific targets to design therapeutics.
Sure, I understand that. So how does funding from the Leukemia Research Foundation help you move along, both with this project and in your career?
Well, first of all, I'd like to say how grateful I am to the Leukemia Research Foundation for this grant. It's tremendous help, especially for a new lab, to receive this kind of funding, both financially and also psychologically, to think that somebody appreciates your potential as a new investigator. Funding is always hard, but it's most important for new labs who are trying to establish themselves in a very competitive field. We need that initial funding to generate new data, which we can use to apply for the bigger multi-year awards and grants. So these awards, they're not small, they're a significant amount of money. So they're really fundamental in helping us get started. So all young investigators are really appreciative of this for these young investigator awards that foundations like the LRF are able to provide to us.
Well, we're certainly very happy to provide the necessary funding for you to move on forward. And how hard is it to get funding for someone who's at your stage, with a new lab and kind of moving forward?
Well, usually in big institutions like Washington University, a new lab will usually start with a help from the university, what's called a startup package, that will help you set up your lab, buy equipment, hire your first few people, and then you start hopefully setting up your research program and generate some data. But it's hard, but it's not. It depends on who you talk to. I mean, the NIH tries to help young investigators. There are specific programs for young investigators or early career scientists. So the criteria are a little bit different for young investigators because they really do see the significance of supporting young scientists.
Right.
Even though they do that, it's not easy. Like less than 10% of grants in general, especially at the NCI, get funded. So it's
the pool is small to begin with, and plenty of young researchers are out there with fresh ideas, innovative ideas, and they just need a little bit of funding to get to move along. And then hopefully with that project, the NIH or whoever will say, hey, you're onto something. We're going to give you $2.5 million or $4 million or something like that, so that you can keep developing that project.
And you'd be surprised how much things cost.
Yeah, yeah. Actually, not anymore. I don't think I'm surprised with what anything costs, because everything seems too expensive.
Well, I find it a little bit entertaining when you have a new student that comes into the lab who's very excited they want to do research, and then they start realizing, I want to buy that thing, I want to buy that thing, and they see how much things cost, and they're like, wow, how can we afford to keep doing this?
Exactly. Yeah, it's always surprising, and hopefully funding from the Leukemia Research Foundation kind of helps with that and doesn't distress young investigators, right away before they even get going, because they can't afford to get the materials they need.
If there's a key experiment you want to do, cost shouldn't keep you from doing it. If it's really important, it's going to help you move forward. So having this funding really helps us not having to worry too much about doing the key experiment and not compromising paying people salaries.
Right. So how did you get into science as a career? What made you want to go into science?
I've always liked science from being a little kid. I would watch a Discovery Channel or something and hear of scientists discovering something new, and that always fascinated me. And it seemed almost a little bit of a bit magical. How could somebody be a scientist? So I was always interested in that and pursued science classes in school and in high school. And at that point, I didn't really have a specific area in mind, but slowly I got drawn into biological systems and genetics, and took a few more genetics courses in college and decided that that's what I wanted to do. And then I got into a Ph.D. program to study molecular and human genetics. I was at Baylor College of Medicine. And that's how, just I guess one thing followed another. I've always liked science. I enjoy it. I still enjoy it. You go to work and you don't know what you're going to discover. You run an experiment and you have a hypothesis. You expect to get a result, and then you get the exact opposite. And that's fascinating to me, trying to understand. I mean, those are sometimes the most exciting experiments, because it's something unexpected, and then you try to figure it out.
Well, fortunately, there are people like you in the world, because while you would find that fascinating that you got the opposite result, the rest of us are ready to throw our computers through a wall because we didn't get what we thought we were going to get.
Well, we have those thoughts too sometimes, but it's like, oh, but that's so exciting.
So that's got you into science. How did you end up picking blood cancers?
So, while I was in grad school, I entertained different possibilities. You do this thing called lab rotations, and you spend a couple of months in a lab that you find interesting. So I spent a couple of months in the lab that did yeast work, and then tried something with bioinformatics. I didn't enjoy sitting in front of the computer all the time. And then I tried something on gene therapy, but that was a little bit slow at the time. And then I rotated in a lab that studied stem cells, the stem cells of the blood. So these stem cells are found in the bone marrow, and they have the ability to self-renew, meaning to make more of themselves, and also differentiate and make all the different blood types, cell types that you see in the bone marrow, the blood, the spleen, and so on. And I found that fascinating, like the function of stem cells, how one cell can become, 20 different other kinds of cells. So I studied the genetic mechanisms of the stem cell maintenance and regulation. But along the way, I kept reading more and kind of got into more of the translational aspect of it, trying to figure out what happens in diseases. And that's how I kind of got into studying leukemias and lymphomas. So when I was moving on to do a postdoc, that's kind of what I was looking for a cancer biology lab that will give me the opportunity to study blood cancers.
Fantastic. Well, we're certainly glad you're in our scientific space. We're all depending on you to find a cure, so no pressure there. Okay. All right. So I have one more question, and this could be perhaps the most important question of the day. If you didn't choose science, what would it have been for your career?
Oh, I will probably be a musician.
Oh yeah? What do you play?
I play the piano, and I've always played the piano. I'm from my family is a very musically oriented family. There's a lot of musicians in the family, and I was exposed to it pretty early on, and I liked it. I enjoyed it, so I started playing the piano from a young age, and at some point I had to choose between being a musician and being a scientist, and I guess science won. But I still enjoy playing the piano. Although I have to admit, I don't have as much time anymore between running a lab and having 3 little children around the house. It's a little difficult to find time.
Yeah, I was about to say, it's got to be hard to do your regular job with 3 kids around.
Especially with virtual schooling. I mean, my daughter is next door doing her Zoom meetings now. So,
how old are your kids?
7, 4, and 2.
7, 4, and 2. So yeah, you've got some good challenge going on there. How do you?
Yes.
How do you achieve? What's one of the secrets of achieving work-life balance, besides not being balanced?
Yeah, there's no balance. There's gonna be it's imbalance. You just have to balance the big picture. It's not gonna be balanced every day. One day it's gonna be skewed one way, and one day it's gonna be skewed the other way. As long as you try to address them all within a bigger time frame, I think it's fine. Mm-hmm. But the way I find balance is I guess clear my head with all these things is to just run. I like running.
There you go.
So I run a lot. I usually run depending when it's hot and humid, either early in the morning or late at night.
I was about to say, in St. Louis, it's humid morning and night. So,
yes. So I've put a lot of miles in the past 6 months.
No, I gotcha. I understand. Well, thank you so much, Dr. Souroullas, for this discussion this morning. I do have a comment online that just says, thank you so much for all you do. We certainly appreciate it. You know that patients and their families appreciate what you're doing. And hopefully this project will end with you finding more good information than you're expecting, and that it moves you on to, just helping to create better treatments for blood cancers. And you never know. If you create the cure, then, the Leukemia Research Foundation could go out of business. And surprisingly, we would really appreciate that. So we hope that you have incredible success. And thank you so much for everything. Okay.
Thank you again for giving me the opportunity to explain my work and reach out to people. And also thank the Leukemia Research Foundation again for their funding and everything that they do for young investigators and for the patients. And yeah.
Thank you. All right.
Right.
We will talk to you again soon. You take care now. All right.
Thank you.
All right. September is Blood Cancer Awareness Month, and we'll be doing several more of these streams. We are going to be streaming with several more researchers during the course of the month. We are going to do a stream with a stream that is designed for caregivers, and we're going to be talking a little bit about COVID-19 and how to, protect your immunocompromised child while he or she is going back to school. how to protect yourself if you are a immunocompromised parent who's got kids who are going to and from school and that kind of thing. So that's coming up. We will also be interviewing a young patient as she navigates her way through a blood cancer diagnosis. So there's a lot more coming up during the month of September. So we hope that you will tune in to our social media channels. We hope that you will go to our website, which is allbloodcancers.org, and click on our Blood Cancer Awareness Month image at the top. There's a toolkit there that you can use to help us spread awareness about blood cancers. And if you have the ability, please donate so that we can continue to help patients living with the disease and funding research so that we can find a cure for these diseases. The Leukemia Research Foundation has been in existence since 1946, and we have spent that time raising money to help patients and to fund research so that we can find the cures ultimately, but on the way, we are learning more and developing, better medications, or helping to develop, better medications and treatments so that people living with a blood cancer can deal with their blood cancer and, eventually be cured from their blood cancer and get on with life. So if you can, please help us out with a donation, and please keep tuning in to Leukemia Research Foundation live streams and other things that we are going to be doing through the month of September. Thanks again for your time today, and we will see you next time.